Hello again, this is Dr. Hannick. This is part C of chapter 11, the PowerPoint lecture of the nervous system. We're going to be talking about the synapse. So the synapse is the nervous system works because information flows from neuron to neuron. So how does the information is transferred from one neuron to the next? Well, it occurs at the synapse, which is a junction between the two. The structure of a synapse. We learned in the chapter on the muscular system, the neuromuscular junction, where essentially it is a synapse between the end terminal of a neuron with the cell body of a muscle fiber. And in between, we have the synapse. So it's the same thing as what we learned already, but this is with regards to within the nervous system. So we have this presynaptic neuron. This is the conducting impulse towards the synapse. It is sending information to another structure. The other structure is called the postsynaptic neuron, in this case a neuron, and the neuron transmitting electrical signals away from the synapse. So it had received information from the presynaptic neuron, and now it is sending this, to send this information to other sites. It could be a muscle cell, or it could be a gland, or another neuron. So here's an image showing you, here's the presynaptic neuron, the nerve terminal here. In between is the synaptic cleft, and this part here would be the cell body of another neuron. So it's communication between two separate nerves. So synaptic connection. So we have the axodendritics, the axosomatic, and less common, the axoaxonal, dendrodendritic, somatodendritic. And then we have two types, chemical synapse, which is predominantly neurotransmitters, and electrical synapses, where we have the ions, namely the sodium, potassium ions, which play a crucial role with generating electrical synapses. So here visually you could see the name is literally the type of synapse. So between an axon and a soma of another cell, it's called axosomatic. Between the axon of one nerve with the dendrite of another, it's called the axodendritic synapse. And here, one of the less common here, an axon with one neuron is communicating with the axon of another, so that's called axonal synapse. So as I stated earlier, chemical synapses, this is the most common type of a synapse process, and these are chemicals called neurotransmitters, and they are found in the axon terminal of the presynaptic neurons that contains these vesicles that are filled with the neurotransmitter. And essentially what's going to happen is these neurotransmitters are going to be released into the synaptic cleft by way of exocytosis. So vesicular trafficking, exocytosis. And then once that happens, the neurotransmitter is going to diffuse across the synaptic cleft, and it is going to then bind to its specific receptor site, which is going to open up electrical channels, allowing the ions, namely the electrolytes, sodium and potassium, to move in and out of the cell respectively. So there are six steps that are involved with this. So the first one is the action potential arrives at the axon terminal of the presynaptic neuron. As a result of that, voltage-gated calcium ion channels open, and calcium floods into the axon terminal, and as a result of this, it causes the vesicular trafficking of the neurotransmitter to be released via exocytosis into the synaptic cleft. So here the neurotransmitter diffuses across synaptic cleft and binds to its specific receptor on the postsynaptic membrane, often chemically gated ion channels. So binding of the neurotransmitter opens up the channels, creating a graded potential. What's important to note that the binding causes receptor proteins to change shape, which causes the ion channels to open. And this causes a graded potential in the postsynaptic cell. And the key thing is it can be an excitatory or inhibitory event. Very important to remember that. Some receptor proteins are also ion channels. So neurotransmitter effects are terminated. How are they terminated? As long as a neurotransmitter is bound to a receptor, graded potentials will continue, so the process needs to be regulated. So in order for you to stop the effects of the neurotransmitter within milliseconds, the termination of its action occurs in three different ways. The first one is diffusion. The synapse, the neurotransmitter simply diffuses away from the binding sites and it is no longer binding, so therefore it stops that process. The other one is through enzymatic action, where an enzyme will degrade the neurotransmitter, becoming inactivated. So therefore there's the other mechanism, and the components of the degraded neurotransmitters will be taken up by either astrocytes or axon terminals of the presynaptic membrane. So synaptic delay is the time needed for a neurotransmitter to be released, diffuse across the synapse, and bind to receptors. And this can take anywhere between 0.3 to 5 milliseconds. So it's really a short period of time. The delay is rate limiting step of neural transmission. So this is the rate limiting step. This is what limits how fast transmission can occur between two different structures. Electrical synapse, less common than the chemical. Neurons are electrically coupled. So these are joined by gap junctions that connect cytoplasm of adjacent neurons. So the communication is very rapid and may be unidirectional or bidirectional. So that's important to know. Found in some brain regions responsible for eye movements or the hippocampus in areas involved in emotions and memory. Most abundant in embryonic nervous tissue. So postsynaptic potentials. The neurotransmitter receptors cause a graded potential that vary in strength based on 1, the amount of neurotransmitter released, and 2, the time the neurotransmitter stays in the synaptic cleft. And depending on the effect of the chemical synapse, there are 2 types of postsynaptic potentials. There is the excitatory and inhibitory postsynaptic potentials. In this case, the excitatory postsynaptic potential, the neurotransmitter is going to allow simultaneous flow of sodium and potassium to flow in opposite directions. A sodium influx and potassium efflux. Sodium influx is going to be much greater, resulting in a depolarization called excitatory postsynaptic potential. Let me pause for 1 second. So this excitatory postsynaptic potential, as you can see, it is going to, as I stated earlier, opens up sodium and potassium gates. The more sodium goes into the cell relative to potassium going out. And as a result of that, you get a positive deflection approaching threshold. This is the threshold here where an action potential would will generate. So this is the stimulus here. You're becoming less polarized, so we're depolarizing here. Now, inhibitory synapse, this does the opposite, but the process is different. What this does is it makes the postsynaptic membrane more permeable to potassium or chloride ion. As a result of that, if potassium channels open, it moves out of the cell, making the cell inside more negative. And if chloride ions open up, chloride ions move into the cell, which also makes the inside of the cell more negative. So in this case, looking at this, it gives you a negative deflection. This is hyperpolarization. We are moving away from the threshold. If we move away from the threshold, then we are preventing or inhibiting to reach the threshold and cause an action potential. So this is how inhibitory postsynaptic potentials work. So summation of postsynaptic neurons. A single excitatory cannot produce an action potential, but you can have a summation where two can be added together, which would lead to that. And inhibitory, postsynaptic potential can also summate as well. So it can do the same thing. Let me show you with a visualization. So here we have temporal summation and spatial summation. Temporal summation is when one or more presynaptic neuron transmits impulses in rapid fire order. So the first impulse produces the excitatory, and before that can dissipate, another one is triggered, adding on top of the first impulse. Spatial summation is when you have postsynaptic neuron is stimulated by a large number of terminals simultaneously from different sources. So here we have an action potential. Here's a stimulus coming in, an axodendritic synapse. Stimulus doesn't reach threshold, therefore no action potential is generated, and we quickly reestablish a resting membrane potential. And then once we did that, we stimulated it again, and we ended up getting the same amplitude. However, temporal summation. The stimulus occurs here. There's depolarization, but still not reached threshold. But if we stimulate again, before it comes back to the before it comes down to the resting membrane potential, then we're going to end up adding more depolarization, which would exceed threshold and cause this action potential to occur. Here's a spatial summation. So spatial is essentially you get the same effect. The only difference is you have two stimulus simultaneously stimulating at the same time, and these two will still act on each other.